freshman class, I'll draw this using stick figure mommy and stuff, and it kind of freaks them out. It's awesome. But I don't really have room to do the whole stick figure baby and stick figure mommy. But basically, when a woman is pregnant, the baby is inside of her uterus, which is a muscular organ, and the muscles are going to push against the baby and expel it through the birth canal. That's kind of generally what happens. All right, so once the baby is ready to be born, there is a flaw in this particular diagram. Does anybody know what the flaw is? It's supposed to be ready to be born right now.
It's upside down.
It's upside down. Yeah, this is a breech, right? It should have, the head should be facing down. So somebody let that slip through. But the head should be facing downwards. And then the head begins to push against the entry to the birth canal, which is going to trigger some stretch receptors. And the stress receptors are then going to send signals up to the brain. And it's going to cause the brain to release a hormone called oxytocin. And what oxytocin does is, once it gets circulating in the blood, is it causes the uterus to contract or push against the baby. All right? So here's where the positive feedback comes in. So you probably know, hopefully, that as the birth process progresses, that the contractions get stronger and closer together, right? In other words, they're amplifying. That's a positive feedback. You change in a certain direction and you continue in that direction. And so as this starts, you're going to start out with some weak contractions, but as the uterus contracts, it pushes against the baby, and the baby's head pushes harder against the opening to the birth canal. That's going to send signals up to the brain and continue to cause the level of oxytocin in the blood to elevate, to the point that the contractions are strong enough to push the baby through. And at that point, what happens? The contractions continue until the afterbirth. People don't like to talk about that part. Right? So then you got to give birth to the afterbirth. And then the contractions will slowly start to level out as the level of oxytocin in the blood begins to come back down. And so the actual elevation as you're giving birth to the baby, that's the positive feedback. Once that's no longer needed, once the baby and the afterbirth have been given birth to, have been delivered, then you're going to go back into a negative feedback, which will bring it back down, essentially. Does that make sense? Your classic example is often blood clotting. So you have a level of circulating platelets in your body. Essentially, this is going to be a simplified version of blood clotting. So then you get a cut, all right, and you start to bleed. And one of the first things you need to do is to try not to bleed, and so the vessels will actually constrict as well. But the platelets will begin to accumulate around the injury, but there's only a few circulating at any given point at any time, and so those platelets are going to send out hormones again, so they're going to start yelling for backup, and that's going to cause platelets from all over your body to start to come to the place where you're injured. And then it's so sweet, the platelets reach out with their little pseudopods, and they connect to each other, and then once they're all connected all the way across the injury, then they'll pull each other tight, and that'll help close up the womb and stop your bleeding. And at that point, once you stop the bleeding and you have enough platelets in place, that positive feedback will stop again. Another example is your bone formation. Bone formation is very similar to making rock candy. Has anyone ever made rock candy? In school? We did it when I was in school. It's where you boil a bunch of sugar in some water, and then you tape a string to the lid of a jar, and then you close it up and you wait for sugar to accumulate on the string. You ever heard of that? That's how you make rock candy. Nowadays, they do things like put Kool-Aid in it so you can have different flavors. We just had plain old sugar. And the first few weeks, you're like, there's nothing on my string. But once the crystallization process starts, then it happens rapidly. And it has to do, in this particular case, with the charges on the sugar molecules attracting more sugar to it. And it causes it to amplify. And that's very similar to what happens with bone formation. So you start out with a cartilaginous model, and then as the ions of calcium and potassium and things start to attach to that calcium, the cartilaginous model, then that tends to accelerate, just like the rock candy. All right. Here's another example. It's not the one I wanted. I'm going to have to go through my phone and find the one I wanted. But this was a dress that they put in the Dead Sea. It's really interesting. But this, at first, it just looks like a normal black dress. But then as the salt began to accumulate on the dress, that accumulated more salt again through the charges on the ions, the sodium and potassium and whatnot in the salts there. So get it to come up. And then if y'all can see it when it comes up. So not as tasty. Not as tasty as. As rock candy. I'll get back to my office. I'll be upset that I changed this. Okay. So this gives you some pictures when they first put it in there. And I think that's the picture that I had or close to it. And they continue to accumulate salt. Look at that thing. Oh no. And then eventually they took it out of the water and they put it in a museum. But that is a positive feedback that did that. All right. Think about building some board owl boxes. You never know what I might be thinking about. Okay. Any questions about homeostasis? Okay, so that's kind of an interlude into something I needed you to understand. All right, so we are going to talk about, I think it's four kind of things that organisms need, all organisms need. So we're going to start with carbon. That's what popped up on this one. We're going to talk about light. We're going to talk about heat, and we're going to talk about water. I think those are the main ones we're going to talk about. There might be another one stuck in there somewhere, but that's the ones I remember. Starting out with carbon. All living organisms need carbon. We are carbon-based. If you remember back, probably depends on who you had for freshman bio again, but maybe in chemistry, carbon has a very specific bonding structure to it. And so it's often drawn like this, right? In your organic chemistries, because you can bond to 4 different things, and so you can link them to each other, form chains. They can also form rings, and then you can attach all kinds of things out here, which are called functional groups and so forth. So they're extremely versatile, and it allows us to build lots of molecules like proteins and nucleic acids and lipids, and which one am I leaving out? Carbohydrates, right? Got to be able to build all those things if we're going to build a living organism. This particular bonding pattern can also be found in silica, but we are not silica-based life forms, right? We're carbon-based. But if you read a lot of science fiction, what you'll see is a lot of times is they do tend to theorize about silica-based life forms. They have them in that. But the reason why is because it has that similar bonding pattern. Right, we're talking about carbon though. All right, so plants are referred to as photoautotrophs. So photo is referring to how they accumulate energy. In the case of plants, they get their energy from light. Auto refers to how they get carbon. And so when you see auto, that means they're stripping carbon from carbon dioxide. And so that's what plants are going to do. They're going to get their carbon from carbon dioxide and their energy from light. We're mostly interested in the carbon part of this in this particular case, but that works really well for them. And that's going to include your plants, your algae, and your cyanobacteria for the most part. Animals, on the other hand, are referred to as chemoheterotrophs. And the chemo part means that they get their carbon from chemicals, which sounds gross because we're chemoheterotrophs. But the reality is that's only our biases in growing up and learning that these chemicals are bad and don't drink bleach and stuff like that, right? But the reality is your food is also made up of chemicals, and that's what it's referring to for us. The hetero part means that you get your carbon from organic molecules. And so the same source that we're getting our energy for the most part. So when you eat a hamburger, you're going to get your carbon and your energy in the same place for the most part. Okay. And so that's going to include the animals, which is what we're interested in, but also the fungi, some of the protists, and some of the bacteria are going to fall into that category. All right. Next slide, super easy stuff too. So as we look at and for whatever reasons, when they talk about plants, they almost always say the whole word, the whole word. Double lectures hard in the first week. The whole word, they'll say photoautotroph. But for animals, it often gets shortened to heterotroph. I'm not real sure why. That's just what happens. But it still means chemoheterotroph. But we'll shorten it as well. We can divide the heterotrophs into two groups. So you have your consumers and you have your decomposers. And frankly, your decomposers are still consumers. It's just that they're specializing on things that are waste or remains. And so waste means pee-poo. Poopé. Anybody watch Ted Lasso? Poopé. And remains means dead bodies. Yeah, dead bodies. But that also includes things like leaves and sticks and anything that's dead, that's no longer connected to a living organism. That's what it means by waste and remains. So that's very specific, and it is a specific group. Can you give me some examples of animals that are decomposers? Usually go bacteria and fungi, right? But there's lots of animals as well.
Worms.
Yeah, your worms, millipedes. What'd you say?
Maggots.
Maggots. Oh, yeah. Did you have something different?
Vultures,
Yeah. Yeah. We tend to call them scavengers for whatever reason, but yeah, that's sure what they're doing, right? For the most part, unless they attack something that's alive still, which they will do if they're injured, not necessarily helpless, injured or helpless. I was reading something about the vultures because we have two types here: the black vultures and the turkey vultures. And when I was coming through school in ornithology, I don't remember learning about black vultures. They were common in other parts of the country already, apparently, but they're growing in population density in the southeastern U.S. And so they were comparing the two groups, and the black vultures are gregarious, which means they hang around together more. And they tend to be a lot more aggressive because of that than the turkey vultures, which are a little bit more solo, not quite as aggressive. And then there's some other differences in the way that they detect food. Turkey vultures have better sense of smell. And so a black vulture could actually follow a turkey vulture to get to some food, for example, and then bully the turkey vulture off of it. Anyway, that was random. So let's look at the consumers. I'm guessing you know the terms on this slide related to the consumers. And so you have the herbivores that eat plants. And you have the carnivores. Carne is meat, so those are your meat eaters. And then you have the omnivores, and that's what we generally are as humans. We have kind of teeth designed for a mix of different types of food, and the physiology that's for a mix of different type of foods. If you're an herbivore, that's fine too. But if you are an herbivore, you have to watch your protein intake and things because of that. So I got a cartoon on the next one. I don't think I gave it to you. So an omnivore and an omnivore. That's what I am, an omnivore. But also, somebody had some company visit them this summer, and the company that was visiting said that they were on a carnivore diet. We're also not really meant to be carnivores either, right? So not our history through time is really more of an omnivore. We eat whatever we can get a hold of, basically. So a mixed diet, ecologically and historically. But it's kind of funny. We run the gambit from, vegan, which is kind of your extreme herbivore, I guess, and then all the way over to people that want to be carnivore only. Then you have the twig and berry ones. I think the twig and berry ones bother me the most because we were never really meant to be just roughage either. We've been cooking and in particular fermenting foods for a very long time. Anyway, none of it bothers me. You could do whatever you want in that regard, but just the fact that they say that that's what we used to do is not accurate, is what I'm saying. All right, herbivory is a problem for animals, including humans. Most animals are not really meant to be herbivores, and so in order to break down very hard to digest plant materials, we often need help. Okay, we need help to do that, which we're going to talk about more on the next slide. And so most of the herbivores, in which that is their complete diet like a cow, are going to have adaptations in order to do that. So herbivory is more complicated, essentially. The reason for that is the cellulose that you find in the cell walls of plant cells. We do not have any enzymes that we produce to break down cellulose. And so a lot of the cellulose that you eat, when you eat a salad, for example, they'll tell you that eating a salad is good for you, which it is. And the reason why is it provides lots of fiber. And that fiber is the cellulose that you're not digesting. It's going directly through your system. But what the fiber does as it goes through your large intestines is it scrapes the walls of your large intestines, which stimulates the production of mucus, and that helps everything move smoothly and quickly through your digestive tract, which is what you want. Okay, so you definitely need that as part of your diet. But the point is, we don't get a lot of nutrition from that. So even though it helps you be regular, right, that's the word for it, it doesn't really give you energy. For example, it doesn't give you any carbon. That fiber doesn't. Right, so kind of keep all of that in mind as we talk about this. This one has a few categories to get more specific about herbivory. Grazer are those that clip off leafy material, so cows are grazers, for example. Browsers, they like to switch over to woody material, and when it says woody material, it doesn't mean biting into the trunk of the tree necessarily, although we do have things like beavers that do that. But more often they're eating the newer growth and the softer things that are coming off the tree. That's going to include like your white-tailed deer, but it also includes the beavers. They're browsers. Then you have the granivores. Notice how that's spelled. There's no I. It's not granivore. It's granivore. And granivores eat seeds. This is a big part when we talk about predation. This is a big part of like the rodent population, for example. And they are what we would call seed predators. So it's because when you eat a seed, you're eating an entire individual, right? And so they fall into that category. And then you have the frugivores. The frugivores feed on fruit. Again, it's frug, not frutivores. Then you have nectivores that eat nectar, like your hummingbirds, and some other insects. You got some of them are eating the pollen. This would also fall into this category. Then you have some that specifically eat on sap, like gliders, sugar gliders, yellow gliders, and sapsuckers and stuff like that. So there's a lot of different ways that you can consume plant materials. All right, and so if you do decide to be on a cellulose-heavy diet, in other words, a plant-based diet, then there are some dietary problems you have to keep in mind. One of the problems that you have is it's low in protein, so you've got to be very careful about that. Eat lots of beans, for example, things that are high in protein, and look for other protein sources. And then because you can't digest the cellulose, you've got to look for things that are a little bit more digestible. And there are plant materials that are more digestible, like fruits. And there is a diet that's fruit-based, although I feel like you would just be loose. All of your stool would be loose constantly if you were on a fruit-based diet. But you wouldn't have any stoppages, I guess. And so there are some things that are easier to digest. To break down the cellulose, you typically are going to have a symbionts, which are going to be either bacteria, fungi, or protists. And usually it can be a community of those things. And so I think you have to help me. Some of you take 140 here. I think in 140 here you might have worked with some termites to see flagellates. Did y'all do that here? They may have taken that lab out. But flagellates are protists that have a flagella. That's what that means. So hopefully y'all remember that much of it. You find those in the gut of termites, and they help them break down the cellulose. In other words, the termites are not eating your house per se. They're just biting off chunks of your house. And then the things in their gut are breaking down your house, right? They can't digest wood without those organisms in their gut. It's the same for the cows, which we'll get to in just a minute. And then what's happening is those protists, fungi, or bacteria, they're releasing the enzymes that are necessary to break up the cellulose. And so cellulose, is made up of glucose. And so you have glucose, and then the glucose is attached to another glucose, and then another glucose is like flipped over and attached, and then flipped over and attached, and there's some special bonds in there. And it's those bonds that we don't have enzymes to break. And so what the bacteria, for example, do is they're able to break those, and when they break those bonds, that releases the glucose that you can now know how to do. Your body knows how to deal with that, okay? If you have that type of thing. All right, down here at the bottom of the slide, this is one of the things that Dr. Hoosman and I have dabbled with over the years, because yellow-bellied sapsuckers, they live in the northern, well, really more up into Canada nowadays, but their historical range would have put them in the northern part of the United States. And during the summer, they excavate small holes in the trees. You see them down here too. You'll see lines of holes, most of the time going this way, sometimes they'll go this way. Those are from the sapsuckers. They can't suck. Birds don't have lips, so they're just lapping it up with their tongue, just to clear that up. But when they do that up north, and they tap into the tree, they tap into the phloem tissue, which is where the sugar's transported, and it leaks. It comes out, and then they can just lick it, right? And they eat that sugar. So that's not a problem. So I studied yellow-bellied sapsuckers down at Mississippi State for my PhD, the winter foraging ecology. And they do make sap wells on the trees in the wintertime. And so I was going to tap the trees that they were using to see how much sap flowed out. 0 is the answer, because they're dormant and they're not doing photosynthesis, and there's no movement of anything in that tissue at that time. So then the question becomes, why are you still making sap wells on these trees in the winter if you're not getting any sap flow, right? And so there's a couple of things that can be happening. Another part of my research was we went out and we girdled the trees. So in other words, we made a ring all the way around it. And when you do that, you cut off the flow of nutrients from the crown to the roots. And that will eventually kill a tree. So I did kill some trees on the National Wildlife Refuge, but I had a permit to do it. But what it does is it doesn't kill them immediately. So the first year, they're going to have a big crown because the nutrients in the roots move through the xylem when they come up in the spring, not through the phloem. The phloem breaks down every winter and has to be rebuilt in the spring. And so that doesn't interfere by removing the soft tissue on the outside of the tree. It doesn't interfere with the sap coming up from the roots to the canopy. But then when it's time and it starts doing photosynthesis and it starts trying to move stuff back downwards, it can't. And so it builds up above the injury. And one of the things I did is I went and I sampled the tissue above the girdle and below the girdle. And above the girdle, you have quite a bit higher of a sugar concentration in the tissue there. It's not going to be running still, the tree's dormant, but there is a concentration of sugars because it wasn't able to go anywhere. Does that make sense so far? For control, I had other trees where I just did a strips all the way around. In other words, I didn't completely cut off the supply. So I have a strip, and then a strip, and then a strip. And in that case, those injuries did not stop the flow. It was able to work around those injuries and get everything down to the roots like normal. All right. And so one of the hypotheses coming out of there is one of the reasons why they continue to make sap wells may be to make better food sources for the future. And so if you go in, and a lot of times they're up in the trees and they'll girdle branches. They sometimes will girdle the trunk, but most of the time they're just girdling branches. Then next year when you come back, you can see that injury, and you know that most likely the tissue above that's going to be sweeter because of that injury. So they're making food for the future, essentially. All right, the other part of that though is even though they may be doing that, it still is not providing enough sugar to support a sapsucker. And so part of my project was doing a whole bunch of math, and I had to look and see what's the energy content of the sugars that you'd find in the sap. And then I measured how much was in the plugs that I took from the tree, how much sugar was there. And then I looked at the energetic demands of the sapsucker itself and what it needed to stay alive for a day. And then I calculated how much of that phloem tissue it would have to eat if it only was able to get the sugar out, if it couldn't do anything with the cellulose. And it would have to eat like several pounds of phloem tissue, which seems unlikely for a bird that needs to be able to fly around. Right? So in other words, it's unlikely it's getting enough sugar by just consuming the sugar in what it's eating out of the tree. And so what's more likely is that they have symbionts in their gut that are breaking down that cellulose and releasing the glucose. And then if you put symbionts into that model, then the amount of phloem that they need to eat per day becomes much more reasonable, essentially. And so that was where my PhD stopped off. And then when I came, because I'm not a microbiologist, so when I came here, I partnered with Dr. Hoosman, and she got interested in the question with me. So we decided to get a student to design an assay that would detect bacteria that can break down cellulose. Okay, and so we had a university scholar do it. We worked on it for 2 years, did a lot of reading. I learned that you can control bacterial growth by giving them only certain foods. And so the media that we were designing had only one energy source, which was cellulose, right? So no sugars or anything else in there, only cellulose. And so if you couldn't break it down, you would die, essentially. But I also learned that it can be really tricky because even though that's our only food source, they still have to have minerals and other things. So we had to have the right amount of metals to mix in there. And so it took us 2 years to come up with the media that we eventually settled on to use. And what we would do then is this was my part of the project. I went out and I collected some termites and brought them back to the lab. And then we popped their heads off. And when you pop the termite's head off, the digestive tract comes with it. And so we were able to take that and they were able to pull bacteria from it. And we spread them onto the petri dishes, which is what you see here. So these are colonies of bacterial growth. And so the way that our assay works is if the bacteria were not breaking down cellulose, then when we flooded it with a certain chemical, and I've already forgotten what we flooded it with, it would have stayed this color. The whole thing would have been blue, all right? In places where the cellulose was broken down, where those bonds were broken down, it turns pink. And so this would be an example of a positive. This one has bacteria that do break down cellulose, essentially. And so along the same times, right at the end of Jody's project, a yellow-bellied sapsucker flew into the library and died. And they almost always call me when that happens to come get it. And so I went over and got it and I said, hey, this one hit today. Is there any way we could check it to see if there's any bacteria in it? And so we just did a very rough thing because we were rushed, it was out of the blue. And so we took a sterile swab and we put it into the cloaca as far as we could push it up into the digestive tract of the bird, and we swirled it around and we put it on several plates. And when we did that, we found a few bacteria that are breaking down cellulose in their gut. Okay? That's as far as we've gotten. We were able to identify some of them. One of them seems to be a new subspecies, maybe a new species, we're not real sure. We haven't been able to classify it very well. We do have the genetic sequence, but we haven't had the time to go the next step and get it named and all that kind of stuff. And the other thing that we don't know is how much they actually contribute to the nutrition of the bird, essentially. So we know that there are cellulose-degrading bacteria in the gut of the sapsucker, but we don't know how much it's actually breaking down food to the benefit of the bird, essentially. Does that all make sense? Okay. And so basically, at the end of the day, so what this is saying to you is sapsuckers are a vertebrate, just like we were a vertebrate. So we do not produce enzymes to break down cellulose. We depend on something else to do that. All right. Probably a more classical example are the ruminants. Can you give me an example of ruminants? Cow, white-tailed deer, cervids in general. All right, and so they have a rumen and a reticulum where the bacteria is. That would be their fermentation point, essentially. And then they have the omasum and the abomasum. Those are the 4 parts, and I got a cute little cow on the next slide. Only one of those is actually the true stomach, even though we call it a 4-chambered stomach. Has anybody ever seen the cows that have the ports on the side? We had a couple on campus for a while, but I don't think we have any currently. So I went to school at Mississippi State for my PhD, and that's a big ag school too. And they had like an ag day, and I don't even remember why we decided to go. I think partly we just wanted to play with Bully, the bulldog mascot, which we did, we played with Bully. But it had a station where they had, I think it's called fistulated. Help me with that. But it's where they put a port in the side of the cow, and you just unscrew the port, and it's big enough that you put a big glove on, and you can put your hand down into the stomach of the cow, and you can move it around if you know what you're doing into the different chambers. And what they'll do is they'll take like a packet of forage, and they'll put it in there, and then they'll pull that packet back out to see how much digestion is taking place. So it's all about the nutrition, what you want to be feeding your cows to build mass and whatever it is they want to do. It's pretty cool, but kind of strange. So anyway, so what the cows actually do is they'll finally clip off the grass, and so that's pretty big chunks. Bacteria can't do much with that, and so they'll swallow it and they'll go into the first stomach. Go ahead and put the pretty cute little cow up there, essentially. And that's going to expose it to bacteria for the first time, and the bacteria will start working along the edges. It can't really do much else. And then the cow's going to be like, and it's going to burp it back up into its mouth. and then it's going to chew it some more. We call that chewing its cud, right? And what that's doing is it's now trying to cut it down into smaller pieces, and then it's going to swallow it again back into that same chamber, and it's going to continue to expose it to bacteria. If it is in small enough pieces, then it can make its way to the next chamber. If it's not in small enough pieces, then it's going to regurgitate it and continue to chew it and swallow it. And then that's how it's kind of regulated. So it has to be small enough to get into this chamber, and then small enough to get in this chamber, and so forth. But really what that's doing is exposing surface area to bacterial degradation along the way. All right? And so that's why they talk about cow burping, because the burping, as they're bringing stuff out of that fermentation point, is going to be methane heavy, essentially, because that's what fermentation is releasing as it's breaking down that kind of stuff. They also fart a lot, also because of the fermentation. I don't have a slide on birds, but most of the time birds that eat plant material also have a fermentation point, but it is pouches that come off the intestines. They're called ceca, or a cecum, singular. And so in a lot of birds, like quail, turkey, things like that are ingesting plant material, if you stretch out their digestive tract, you'll see these long pouches, and that's where the fermentation takes place. One of the weird things about the sapsucker, the reason why we don't know how much the bacteria contribute to their digestion, to their actual energetic needs, is because they don't have those pouches. In other words, we don't know where the fermentation point is, or if the stuff is staying in there long enough for fermentation to take place and contribute, essentially. It's just weird. It's a weird situation. There is one species of bird that is a foregut fermenter, meaning that it does fermentation before the stomach rather than after the stomach, and that is the Hoatzin, which is a really weird bird. Has anybody ever heard of a Hoatzin? The Hoatzins, when they hatch, they still have claws on their wings, and they can claw up trees. They lose those claws when they become adults, though. They're really weird. They're big birds. They live down in South America, in the Amazon. And they're also the only foregut fermenter in the birds, so that's a little unusual. I guess, technically speaking, cows are going to be foregut, right? Because the true stomach is the last pouch before it goes into the intestines, so they're foregut fermenters.
Do we know why that specific species is foregut fermenters?
I don't know about why per se, but they're using, if I remember correctly, they're using the crop, which is that storage organ, which would make sense to put it there and ferment it, and then pass it through your digestive tract. And so then you could ask the question, why are the others not, right?
Mm-hmm.
So I don't really know. Probably partly because they are big. They're not turkeys. They're in a completely different group, but they kind of have that turkey shape. They're like round. And so it probably has to do with flight itself. And so being a foregut fermenter probably isn't conducive to being a flighted bird. Which they are flighted, but they're not flying much.
Okay.
All right. So we do have some herbivores that are not ruminant. So I guess I kind of jumped the gun there on those a little bit. They tend to have simple stomachs. They don't have four-chambered stomachs, but they have long intestinal tracts. And so I kind of already covered the turkeys. Not only do they have a long intestinal tract, but they've got pouches on their intestines for fermentation. And so the fermentation point for our non-ruminant herbivores is the intestines, somewhere along the line in the intestines, essentially. So post-gut fermenters. All right, I want to get one more slide, I think. Yeah, let's finish up the herbivores. So your non-ruminant herbivores would include not only those that have the long intestines, which I think is a very common strategy. That's what the birds do. But it also gets those that eat their poop. Right, so that's coprophagy. That's the fancy word for it. Copro means poop. Poopé. All right. Phagy means to eat. So to eat your poop. And basically, it's the same strategy as the cow. We just don't think of it as the same, right? Because what they're doing is they're doing a coarse digestion, incorporating the bacteria and pooping out that pellet, and then eating it again, which is going to help you break it down even further, get more energy out of it with a second fermentation of it, and then the second time you poop it out, you don't eat it. Okay, and the rabbits know what to do. So here's some pictures of some rabbit feces, some cecotropes and hard feces. And I forget which is which, but they eat, I think they eat this one. They might have rabbits that help me remember, because it is weird. And then I think those are the ones they don't eat. Yeah, any questions about eating your poupée? Then we're ready for carnivores, so we made it with 2 minutes to spare. Any questions about being an herbivore as an animal? We were very focused on animals and how they have to deal with being an herbivore. Okay, the next time we're going to talk about carnivores, and then we're going to move off of carbon. I think heat's next probably. We'll talk about how do you manage your heat balance. Oh, I'm probably going to go to the gym.